My name is Dr. Mervyn Campbell. Now we're going to deal with laboratory practical number 7. And the objective is to measure the erythrocyte sedimentation rate, or the ESR. And just a little preview of it. It's a non-specific test that measured the degree of inflammation in the patient. And it could also be used to monitor the response of the patient to therapy and the progression or the remission of the disease. It is not a specific test. It does not indicate what type of inflammation is present, or what organism or what external agent is causing the inflammation. It measures the distance the red blood cells will fall in a specifically made vertical tube over a given time period, and is usually reported in millimeters per hour. The length of time is usually 1 hour for the procedure. There are several conditions which may result in an increase or a decrease in the ESR, and some factors are like the red cell shape and size. A very small red cell will tend to sediment out a lot more slowly. So things like erythrocytes and sickle cells and acanthocytes and so on, they tend to sediment out more slowly, and therefore over the 1 hour period the column of sedimentation would be high. In the case of increased plasma proteins, particularly things like immunoglobulins and fibrinogen, they tend to sediment out a lot more quickly, and therefore the column after sedimentation will be lower, and the cells, the time that it takes to reach sediment will be a lot quicker. We also have other factors that may influence it, and the increased viscosity of the blood. Like for instance, if you have a very high immunoglobulin count or level, that may cause a decrease in the rate of sedimentation. There are specific kits that are used. However, these kits are now replaced by an automated method that are used in some of the more advanced labs. However, the method that is used is called the Western, or the AutoZero Westergran erythrocyte sedimentation rate system. And we have with us Ayuka King and Ferlene Santiago, who would be demonstrating the process to you. When this is done and is presented to you, we will I'll discuss it, and it should be placed on e-learning, and I'll discuss it with you at that point.
Okay, so this is the erythrocyte sedimentation lab. We have the kit here, which is the Globe Scientific, the Western Grand ESR system. Okay, so today we have 2 samples, sample 1 and sample 2. And I've labeled the ESR tubes, sample 1 and sample 2. Basically, we draw our EDTA sample, whole blood, because that's what we're going to use. And then we transfer the sample into these tubes, 1 ml. So these are what the tubes look like. I'm going to transfer it. First we invert our samples, mix, and we transfer them into our vials. 1 ml. There's a reference mark on each tube. You can see the reference mark. Okay. Okay, so there's the reference line that we filled. Exactly 1 ml of the whole blood sample in the tube. Then we invert the tube 5 to 6 times. Okay. We have our second sample. And we invert our samples 6 times. It's important to gently tap our tubes on the bench to get rid of any blood that is stuck up inside the caps. Then we place our measuring device. Hold one sample firmly on the bench in one hand and our measuring device in the next. Firmly grip the sample while placing the device down slowly until it reaches the 0 mark. Then we set our timer for 60 minutes, 1 hour.
Observe that the lab technician did not forcefully push the pipette down into the sample tubes. But if you push it too aggressively down, the blood can come right up the pipette tubes and splash even in your face or the hands or on the counter and so on. So once you puncture the stopper of the tube, once you puncture it, gently push it down into the sample. Don't aggressively push it down.
Okay, so our 60 minutes has passed and we're going to read our ESR. You can look closely, you can be able to read it. The lower it gets is the higher number. So in this case we have a reading of 44 mm per first hour. That is our units to read our ESR. Second sample is around the same, 44 mm per first hour. So there you have it. This is how to set up a erythrocyte sedimentation rate, and this is how to read it after 60 minutes.